Sairam, everyone. Sairam. We now have presentations by the research scholars of Department of Chemistry. I request the research scholars to stick on to time. The time limit is 4 to 5 minutes for each presentation. Thank you. Not going back. In case this like this now. This way. Om Shri Sairam. My name is Undavalli Venkata Gopi. I'm a final year research scholar from Department of Chemistry. I'm working under the supervision of Dr. Kumar Sai Smaran. Our lab mainly focuses both on energy storage and electrocatalysis. On the energy storage front, today I'll be talking about supercapacitors. This is the first chapter of my thesis. And since most of the basic things have been discussed earlier by Venkata Sharda ma'am, I'll be going directly onto my research topic. Next. Yeah. Previously. This is the summary of my research where I have worked on defect-based materials. I engineered defects in the materials to both applications like energy storage front and also the electrocatalysis front. Today I'll be telling you about one chapter of my thesis which focused on defects for supercapacitors and how can we use these defects to our advantage for using supercapacitors. And this is the Ragon plot which ma'am has showed. Basically, supercapacitors come somewhere in the middle where they have very minimal energy densities but higher power densities. Power densities meaning they deliver a high amount of energy in a shorter amount of time. So supercapacitors come under the there in the Ragone plot. Next. I should keep blank. If you see the mechanisms involved in a supercapacitor, yesterday ma'am was discussing about EDLC supercapacitor, where the charge storage is only electrostatic, there is no redox reaction which is happening. And the second case is the pseudocapacitor, where a redox reaction governs the charge storage. And the third would be a hybrid of both of this, where you use a EDLC electrode on one side and redox based electrode on one other side to improve the energy densities of supercapacitors. As I've told, I focused on material defects. These defects range from 0D to 3D and they are categorized accordingly. I'll be talking about point defects. These point defects are categorized mainly into stoichiometric and non-stoichiometric. Stoichiometric being if we have a material and if the ideal cation to anion ratio is retained and if still the material is defective, that's called a stoichiometric defect. And if the cation to anion ratio is altered, the defect is termed as non-stoichiometric defect. I'll be talking about non-stoichiometric defect. One particular example that I've used in my research is a perovskite oxide of the structural formula AbO3, where for example, if we take an LaNaO3, A is La, lanthanum, B is nickel and oxygen. Because nickel tries to exist both in +2 and +3, there is an inherent oxygen defect existing in this material, which I showed here. In addition to the pseudocapacitance, we can also use these defects for realizing the an additional storage of charge. So that's what we're going to see here. So what we try to do is use these defects also for charge storage, because in traditional supercapacitors only the surface of the material is used for charge storage, and often the bulk of the material remains unutilized. So to utilize the bulk of the material, what we try to do is create defects in the material so that electrolyte can penetrate into these defects, and the bulk of the materials can also be utilized for the charge storage. So that's the hypothesis. And we used a OH minus, the hydroxide electrolyte, to actually occupy the vacancies in the material and then store the charge. So the idea is, if we can increase these defects somehow using some strategy, so that we can actually store more charge compared to the non-defective material. In that pursuit, we actually took a precursor and tried to anneal in nitrogen and air. Air annealing should give us lesser defects, and we thought nitrogen annealing should give us more oxygen defects, because of the lack of the oxygen in the atmosphere. With that hypothesis, we went ahead, but what we observed was a formation of two different phases. Air annealed samples gave a phase of typical LaNiO3 perovskite phase, but nitrogen annealed samples gave us a different phase, which is a derivative of perovskite, which is actually excess in oxygen. If you see the structures here, this is LaNiO3 where there is a defect of oxygen, but when you see a derivative of perovskite oxide, because oxygen can penetrate through the latter, through the layers also, there is an excess of oxygen in the other structure, derivative perovskite. These two, to test the hypothesis whether oxygen vacancies actually help in charge storage, we did a cyclic voltammetry study to see whether the defects actually play a role in the charge storage. These two are a comparison of a defective and an excess sample, where we can see, uh, if we calculate the specific capacitances, it is almost half of the order. The air annealed samples which are containing oxygen defects gave us 100, and nitrogen annealed samples which are excess gave us only 59. And these defects and oxygen vacancies are also characterized using various titrations. As sir was asking yesterday, actually we characterized the defects using iodometry titration, where we used the oxidizing nature of Ni3 plus. Na3 plus gets reduced to Na2 plus. It oxidizes I minus to I2. So we can use this to quantify nickel 2 plus and 3 plus oxidation state, and thereby we can back calculate the oxygen stoichiometries. So this we have used for calculating the oxygen defects in a material, and also we used XPS as well. This is one of the hypotheses where we can use defects in a material for utilizing the bulk for charge storage, and we have done many other works also, uh, wherein we composited it with the carbonaceous materials ranging from 0D to 2D, 0D to 0D being graphene quantum dots, radium graphene oxide, and 1D being carbon nanotubes. So wherein we observed an increase in specific capacitances or the charge that is stored, because the bulk of the material is being utilized now. And the other part of the research is with this layered perovskite oxides, where they act as a very good electrocatalyst for oxygen reduction reaction per se. And also we have used nitrogen doped activated carbons to composite with these things to improve the onset potentials of oxygen reduction reaction. Anything further doubts or questions regarding my research, you can reach out to me, and thank you for your time.
I just have a very quick question. So, in your two phases where you see So in the nitrogen atmosphere, you are getting a non-perovskite oxygen-rich
Yes,
phase,
yes, sir.
whereas the air atmosphere, you are getting the
Mm.
ABO3 phase.
Yes, sir.
Whereas I would expect the other way around, because in air there is more oxygen, so it will lead to a oxygen-rich phase, whereas in nitrogen atmosphere it would
Mm.
lead to a.
But exactly
But
what
you are
is
getting the other way around. That's my question.
correct, sir? Exactly what is happening? The precursors are lanthanum is in +3 state and nickel is in +2 state during the synthesis. In air atmosphere, there is sufficient oxygen where nickel gets oxidized to 3 plus and there is a formation of LaNiO3 phase. But in nitrogen atmosphere, there is no sufficient oxygen for the oxidations to happen, and there is a retention of that oxidation state. That's why there is an La2NiO4 phase which is formed.
Fine, thank you.
Sairam, Sairam, my name is Anil Ram. I'm currently in third year and I'm working under Swayam Siddhakar and Dr. Lakshminath sir. Next slide. Uh, I'm going to present one of my research topic, that is drug repurposing. Drug discovery process consists of the following phases which we can see here. Further, this phases has sub-phases due to which to develop a drug from discovery to approval it takes a time of approx 8 to 15 years, and average time varies, but estimates range about 4.46 billion US dollar at the higher end. As we have seen, recent pandemic, that is COVID, there was urgent need of medication, but to develop a drug, it takes almost 8 to 15 years. So one of the way to develop a drug is drug repurposing. Drug repurposing is a method in which existing drugs are repurposed to identify new therapeutics, and it is a recent method that has been emerged in article in 2004 by Asborn and Thor. Drug repurposing addresses unmet medical need. It also addresses orphan diseases and emerging conditions, like leveraging side effects of existing drugs as it has unexpected benefits, and repurposing based on observed effects. Drug repurposing has advantages over traditional drug discovery as it skips discovery and phase 1 clinical trials. Drug repurposing is less time consuming, less risk of failure, safety profile and clinical efficacy already exist. And drug repurposing, this slide shows the methods of drug repurposing, where drug repurposing are significantly faster and less expensive compared to traditional drug discovery, as existing drugs existing drugs as safety profile and are FDA approved drug. There are main two methods: activity based or experimental method, and in silico or computational method. Here, activity based or experimental method. In this method involves testing existing drugs against new targets or disease model. This can be done in various ways. In image you can see, like cell-based assay, animal studies, or clinical studies. Next one is in silico or computational method. This method uses computer to identify potential new uses for existing drugs. This can be done by analyzing large data set of protein-ligand interaction, disease gene association, or chemical structures, as you can see in the image. Drug repurposing can be sustainable in terms of cost benefit, like it has cost reduction, utilizes existing drugs, time efficiency, like it skips phase 1 clinical trial, accelerates availability of treatment. Other advantages like safety profile already exist, reduce risk associated with developing entirely new drugs, faster evaluation in clinical trials. The last 2 points, that is synthetic procedure and economical, are for in silico methods over in vitro, as many drugs have established synthesis procedure and hence manufacture can be done easily. Existing drugs are economic to use as they are already in circulation. These are the success stories of drug repurposing. As you can see, for example, minoxidil. Originally, it has been used as a treatment for angina, that is chest pain, and it has been repurposed for tropical treatment of hair loss. Another example is metamorphin. That has been developed in 1950s for type 2 diabetes. Recent research suggests that it is also beneficial for certain type of cancer and also for neurodegenerative disease like Alzheimer's. Pasconazole and ravconazole are the drugs for Chagas disease are currently in clinical studies. We have seen COVID-19 that drug repurposing has picked up pace. As we have seen, there was rush to repurpose existing medication for the pandemic. These are the 8 targets. These are the latest 8 targets that has been selected for protein ligand interaction studies. These are the 60 ligands that are available in our lab and are pure and approved API drugs. These drugs consist of different class of drugs such as antibiotic, for example, penicillin G, antiparasitic, for example, albendazole, mebendazole, anti-inflammatory like ibuprofen, norfloxacin, anti-malarial like chloroquine phosphate, etc. Molecular docking has been carried out for the 8 targets with 60 ligands, and based on binding affinity, novomycin, omeprazole, and loperamide has been found common across Swissdog and Autodog, whereas neomycin bisulfate has shown highest binding affinity in Swissdog, whereas mebendazole is found common in Autodog. For this reason, following 5 ligands has been undertaken for further protein ligand interaction studies. This This is the result of ensemble docking, which that has been carried out for protein 3v4O and novomycin. This is the ensemble docking for protein. For 20 nanosecond, it has given 200 frame, and ligand here used is novomycin. Next. In conclusion, this receiving expertise and advice in these areas will help provide critical techniques, methodologies needed for research. There are softwares like GrowMax, Schrodinger, Charm GUI. And overall, this advancement will contribute to progress my PhD and potentially lead to significant development in the treatment of drug repurposing. Thank you.
Om Sri Sai Ram. I am Manohar Bujel from Department of Chemistry. I am working under Professor G. Nageshwar Rao sir, and our lab focuses on natural product. It's we semi-synthesize this natural product into various derivatives and checked its anti-cancer properties. So today I'll be talking about isolation and separation of arginolic acid from acetic acid and synthesis of amide derivatives of arginolic acid and investigation of their anti-cancer activities. So as we all know, cancer is the most deadliest disease that the mankind is facing, and after having the more advancement in the medical field, though the challenge of treating cancer still remains. And the major challenges for the treatment of cancer is the heterogeneity of the diseases, toxicity and side effects of the therapeutics that are given. There are limited biomarkers that are available to target. There are lack of molecular diversity, and the last one is the drug resistance. Now, coming to arsenolic acid, this is the pentacyclic triterpene acid that was first isolated by King et al. And this arsenolic acid is of more it has a various biological activity. And my problem was to isolate this arsenolic acid and separate this arsenolic acid from acetic acid, because this arsenolic acid, though it has more of biological activity, it in all the sources it comes along with its isomer acetic acid. So it is very difficult to separate these two acid here. So you can see this is the basic procedure that I followed here. So from terminally Arjuna, I took the heartroot of terminally Arjuna, crushed it, and through it I refluxed it with ethyl acetate here and got the crude extract. And these crude extracts were passed through column chromatography using chloroform and methanol. We did normal chromatography, and with this ratio I got 3 compounds, that is compound A, B and C. And compound, we characterize these compounds here. So for the characterization of compound A, we did mass spectrometry LCMS we did, and we came to know that this compound was the mixture of dihydroxy acid, that is corosolic acid and macellinic acid. Coming to the characterization of B, we again did LCMS mass, we recorded the mass and came to know that this compound is also the mixture of arsenolic acid and acetic acid. Now coming to the characterization of C, now we again recorded LCMS and found that this compound is a tetrahydroxy acid. And after further purification, after further characterization with 13C NMR and 2D NMR, we came to know that this is the terminolic acid. And this was the first time it was reported from the terminalia Arjuna. Now coming to the separation of arsenolic acid and acetic acid, if you see here, there is only the difference is only the position of CH3 here. If you see here, in the case of arsenolic acid, the two methyl groups in the geminal position, and here if you see it is in the adjacent position here. So their RF values are same, so it is very difficult for us to separate this arsenolic acid and acetic acid. Now if you see the 13C NMR of this arsenolic acid and acetic acid, if these are present there in the compound, you can see these 4 peaks here, and there are two peaks here. So this, if you can see these pink peaks, pink colored circled peaks are of the acetic acid, and the dark black circles are of arsenolic acid. So we developed a method, and we successfully separated this arsenolic acid and acetic acid. Now this is the summary of my first part of the talk, that is from terminally Arjuna we were able to isolate the mixture of mixture of this corosolic acid and macellinic acid, and the highlight of this work was we were able to isolate 11.5 gram, that is 1.5% of arsenolic acid, and 2.5 gram of acetic acid from 1 kg of heart of terminally Arjuna. And just to give a perspective, if you look at the market value of arsenolic acid, it cost around for 1 mg it cost around 32,000 rupees, but we were here with a less amount of 10,000, we were able to get 11.5 gram of arsenolic acid. Now once we had the arsenolic acid, we went to made different amides of arsenolic acid, and here is the scheme here, and this is the characterization of amides here, and all these 23 amides were sent for their anti-cancer activity preliminary in the NCI, and these starred ones were selected for the higher dose activity. Now this is the compound which was most active here, that had the amide of 1,4-bipyridine amide here, and this was the MTT assay that I have done, and we got the IC in the CT26 colon cancer cell line, we got the IC50 of 1.88 micromolar here. And this was, again, we wanted to check the mechanistic study of compound T1,14-bipyridine, that is in 2L, that is in the CT26 and HCT116 here. And we did PI uptake assay, cell cycle assay, ROS assay, apoptotic assay, and animal studies. And in HCT116 colon cancer cell line, we did MTT assay, PI uptake, cell cycle, and ROS assay. Now, this is the result of PI uptake assay. If you can see here, in the untreated one, that is in the normal case, there is no PI uptake by the cells here, but in the case of 12 treated cancer cells, here there are more, the PI stained cells are here in both the cases. And in case of cell cycle assay, if you can clearly see here in the CT26 and HCT116, our compound was able to arrest the cells in G0, G1 phase here. And this is the ROS assay. that I conducted for this compound, whether to check whether to see the mechanism as to what our compound is able to do to the cells. Here you can see in the untreated there are no ROS cells, but in the case of 2L treated you can see clearly there is more population of ROS cell is here. And I went to check the apoptotic assay, and you can clearly see there are 20% of the cells are in the late apoptosis, and 70% or 60% of the cells are in the late early apoptotic phase. So we went on to again check this compound in mice model, and as you can see here, this is the procedure that we followed, and this was the result here. In after 21 days we were able to see that our compound is able to inhibit the growth of tumor in the mice by half.
Sairam.
Yeah, this is my summary of the talk, and credit and acknowledgement, and thank you. If you have any questions, you can talk to me in email, or you can email. Thank you.
Thank you for your very nice presentation indeed. You isolated a very important triterpenoid acid, arginolic acid. If you look at the structure of this particular natural triterpenoid acid, you see that at 2 and 3 positions it has hydroxy group, okay?
Yes, sir. Yes, sir.
And 2 hydroxy is alpha,
Yes, sir.
3 hydroxy is beta.
Beta.
So nature selected in this particular fashion.
Yes, sir.
Do you have any idea behind this? Why nature goes for this type of selection?
So that I'm not sure why it selects particularly this.
Now, do you understand my question?
Yes, sir, I got it.
So it's very easy. If you look at this structural pattern of your cyclic system,
Yes, sir.
both the hydroxy group at the equatorial position.
Yes, sir.
Okay.
Yes, sir.
So you know that equatorial
Position is
subposition is more
Yes,
preferred.
sir, preferred.
So what's about the NMR, proton NMR of your carbonyl
Okay.
protons
Sir,
at the
carbonyl.
3 positions, not 2 positions?
Yes, sir. I will check.
Can you understand the stereochemistry of 3 OH? Whether
sir.
it is alpha or beta, it can
Beta
be
it
very much ascertained from the proton NMR
sir,
space.
yes.
What's the clue? Now, you just tell me
Yes, sir.
whether the alpha hydrogen
Yes, sir.
at C3 will be upfielded or downfielded.
Sir, it will be little bit upfield toward downfield. Sir, it will be downfield. Up.
Downfielded if your hydrogen is in alpha position.
Wait, sir. Yes, sir. Change the slide.
It's okay. It's okay. You characterized it very
Yes,
well.
sir.
So this is the clues actually. You will
Yes,
be
sir.
faced this type of questions in the interview.
Yes,
So I think it's a very, very interesting work,
Yes,
and it is obviously praiseworthy work, no doubt. Thank you.
Om Shri Sai Ram, offering my humble pranams at the divine lotus feet of Bhagwan. Today I'll be presenting a small part of my research work, what I'm doing here. So my work is design of novel materials of PVC blades for blood bag application using multi-scale molecular dynamic simulations. Okay, the motivation of my work is like the blood which is stored, the blood bag which is stored. When you store the blood for more than 42 days, we cannot use it. The shelf life of blood is only 42 days. And the blood bag is made up of PVC polymer and DHP plasticizer. So the advantage of this DHP plasticizer is reduces RBC hemolysis. So the shelf life is increased. So but the disadvantage is it's a carcinogenic and endocrine disruptor. So the DHP is banned. The Europe DHPs they're planning to ban this DHP plasticizer by 2030, and they want to get some other material for this. So my study is to replace this DHP plasticizer or get some new material for blood storage applications. So I'll be doing what I've done is a computational study for these plasticizers and polymer materials. So one of my work is like I'm studying the polymer plasticizer interaction, a one-on-one interaction between the polymer and the plasticizer using molecular dynamic simulations. I'm using umbrella sampling technique and getting the potential of mean force values for the polymer and the plasticizers of different polymer length scales and the different set of plasticizers. I chose this plasticizer these are the set of plasticizers which I chosen for my study. All these plasticizers were taken from different literature sources where it was given it has a potential of replacing DHP as a plasticizer. So I did 30 independent simulations of this calculation to get the overall picture of my simulations. So we can see here, so this is the PMF value which I get for one-on-one interactions. I'm taking these values and averaged over 30 simulations. And for each polymer plasticizer interactions, like for one polymer with different plasticizers or one plasticizer with different set of polymers, I'm getting this plot, PMF plot, I'm then normalizing it. So with these values, I'm constructing a model where I can understand the type of interaction between various set of polymers and the plasticizers. One of the set of plasticizers, similar type of plasticizers are orthophthalates. So in this, the only difference between this five plasticizers are the only the chain length is increasing from the DIBP to DITP. So increasing chain length from 4, 8, 9, 10, and 13. So what I see is so when I change the polymer length, so for different these are polymer DI these are polymers, orthophthalates, molar mass of orthophthalates, and interaction with different set of polymers from PVC 6, 10, 14, and 20. When I see the change in the polymer interactions, I can see the change in slope from negative to positive. So with like both polymer and plasticizer doesn't have any ionizable groups. The only possible interactions are like through van der Waals or electrostatics. So for longer polymer chain, we have seen the it is positive slope. So with other studies, we got to know this like it's kind of van der Waals interactions. And if you plot the slope values separately with molar mass of polymers, we can see the increasing in the slope values from 400 to the PVC 6 to PVC 20. Same if you like extrapolate it, it's not going to zero. So there is some other interaction which is possible, that is the electrostatics. So if I put this in the fit it in the slope, I can see this for slope it is 1 by r, and so 1 by f, one is exponential and is 1 by r. So I can understand the polymer plasticizer interactions. I did for a different set of plasticizer families for understanding this type of interaction, and we developed a model that where we can predict the type of interaction between the polymer and plasticizer. One of the example is yeah, next one. So these are for different plasticizer. I got different slopes and intercept values. Just short of this. Next one. So here we can see that I have two sets of plasticizers, DHP and DHT. This is 1, 2 substituted and this 1, 4 substituted. I can differentiate between these two plasticizers by seeing the slopes and intercept values, like the van der Waals and interactions. By this, I can with this, if I develop the model more, I can get the prediction of the plasticizer systems. So similarly for other branching effects, the ring substitution effects, and all those types of studies are done. So other thing is like I rank the plasticizer depending on the type of interactions. So the stronger the interaction, the stronger the bonding. So less the leaching of the plasticizer from the blood bags. So we found out that the TOTM, DHP, BTHC, ATHC are the top strong plasticizers, and the literature from the other experimental literature also supports our results. So from this study, like initial studies, we were able to develop a model where we can analyze the type of interaction between this polymer and plasticizers, and we are trying to extrapolate with other set of polymers which are possible. So till now, what I did was only a PVC polymer, and uh, we found out that TOTM had the best, strongest interaction between the polymer chain. So and also our results were supported by some of the experimental results from the literature. So when we rank the plasticizers TOTM, BTHC, and we are using this selected plasticizer for my further studies using atomistic MD simulations and the coarse grain simulations. Okay, that's all. Thank you. Okay, one more thing. I want to thank my supercomputer facilities for the server they're providing. Thank you.
Sarah, thank you, research scholars. We now have concluding remarks by Mr. Venkatagopi, research scholar, Department of Chemistry.
Sairam, it is my honor to deliver the concluding remarks for this two-day symposium on chemistry for a sustainable future, bridging science and society. The discussions and deliberations we have experienced over these 2 days have been both intellectually enriching and deeply inspiring. Let us remind ourselves of the sequence of events that unfolded over these two remarkable days. The symposium began with a spiritually uplifting invocation through Vedam chanting, followed by the ceremonial lighting of the lamp by our Vice Chancellor, Head of the Department, and distinguished speakers. Our Head of the Department of Chemistry, Professor Rajini Bandari, provided a thoughtful introduction to the symposium's theme, emphasizing the transformative role that chemistry can play in addressing global sustainability challenges. The Vice Chancellor, Professor B. Raghavendra Prasad, in his address, inspired us to actively implement principles of sustainability such as decarbonization and also fostering interdisciplinary collaborations. He highlighted the importance of integrating sustainability into every facet of our actions and emphasized that many of the events planned as part of Bhagwan Sri Sathya Sai Baba's centenary celebrations in November 2025 will reflect this ethos, ensuring that sustainability is considered in every activity. Sir advised us to apply the ideas discussed in the symposium to create tangible outcomes. Sir's words set the tone for the profound discussions that followed. The technical sessions on day one of the symposium included a total of 5 insightful talks. Professor Gautam Brahmachari began the day with an insightful session on the principles of green chemistry. He showcased green matrix optimization techniques in organic synthesis, highlighting electrosynthesis as an energy efficient alternative for creating desired products while adhering to the principles of sustainability. Professor K.R.S. Chandrakumar shared valuable insights from theoretical chemistry, demonstrating how computational methods can reveal important details of chemical processes that are often challenging to observe experimentally. His examples on hydrogen generation and ammonia synthesis have revealed how material curvature, aromaticity, and charge transfer can enhance catalytic efficiency, and how confinement approach can ease the process of ammonia production from nitrogen. Professor Venkatakrishnan presented cutting-edge work on sustainable synthetic routes focusing on biomass valorization, carbon dioxide conversion, and mechanolysis of PET plastic waste. The discussion on quantification of green metrics and innovative catalytic approaches exemplified practical applications of green chemistry. Post-lunch, Professor Vinayak Sinha shifted our attention to environmental chemistry. He discussed the impacts of greenhouse gases, volatile organic compounds, and secondary organic aerosols on global temperature rise and air quality. By emphasizing evidence-based monitoring and chemical fingerprinting, he provided strategies for targeted interventions to improve air quality. The day concluded with a talk from Dr. Venkata Sharada, who provided a comprehensive overview of energy storage technologies. She discussed the potential of combining batteries and supercapacitors and emphasized the need for sustainable materials. She also shared her work on recycling spent lithium-ion batteries and using biomass-derived electrodes for EDLC supercapacitors and asymmetric supercapacitors. The final segment of the day was a presentation by a representative faculty from the department showcasing the research activities and advancements within the department. This session offered a glimpse into the ongoing efforts and achievements of our research groups. Day 2 of the symposium began with a thought-provoking panel discussion featuring distinguished experts. The panelists shared strategies for integrating sustainability into academia, research, and daily life. Their practical ideas and inspiring perspectives provided actionable insights for everyone present. Following the discussion, we had a total of 3 technical talks, first being Dr. Vishal Ketan opened the technical talks with a presentation on surface solutions as enablers for sustainable manufacturing, illustrating how surface engineering innovations are transforming industrial processes for greater sustainability. Professor C. Subramaniam delivered a compelling talk titled Swami, Sun and Sustainability, drawing from the Vedas and the teachings of Bhagwan Sri Satya Sai Baba. He highlighted how ancient knowledge aligns with modern sustainability practices. His work on solar thermal converters and water purification using nanocarbon fluorides demonstrated how scientific innovation can play a crucial role in addressing global challenges. The technical talks concluded with an engaging session from Dr. Sampath Kopole on frontiers of AI research. He illustrated how artificial intelligence is driving innovation and sustainability in healthcare and life sciences, enabling more efficient resource use and impactful outcomes. The post-lunch session brought out a vibrant display of creativity and talent. We have student clubs presented live demonstrations showcasing practical applications of sustainable practices in chemistry. Their enthusiasm and ingenuity were a testament to the promise of the next generation of scientists. This was followed by a poster presentation by undergraduate and postgraduate students covering a wide range of topics related to sustainability through chemistry. These presentations offered an opportunity to appreciate the diversity of ideas and dedication of our budding researchers. After the tea break, the spotlight shifted to our senior researchers, including myself, who delivered concise presentations of our ongoing research within the department. Across these 2 days, the symposium has demonstrated the profound impact of chemistry in addressing sustainability challenges. From green synthetic methods and advanced theoretical models to environmental monitoring, AI-driven solutions, and energy storage innovations, the discussions have underscored the transformative power of interdisciplinary collaborations. Equally inspiring was the integration of ancient wisdom with modern science. As Professor Subramaniam reminded us, the principles of sustainability are deeply rooted in the Vedas, offering timeless lessons for creating a harmonious and a sustainable world. Finally, the contributions of our students and scholars highlighted the sustainability is not just a concept, but a mindset, one that must be nurtured at all levels of education and research. As we move forward, let us remember that sustainability is not just a goal, but a continuous journey that requires collective effort. Let us take the lessons we have learned and transform them into action, whether in the lab, in the classroom, or in our communities. On behalf of all the organizers and all the participants of the symposium, I extend heartfelt gratitude to our distinguished speakers for their invaluable contributions. We wish you a safe journey back to your destinations and look forward to seeing you in our future gatherings. Sairam, thank you.
Thank you, Mr. Gopi. Now we have vote of thanks by Ms. Manaswini.
Om Shri Sai Ram, respected dignitaries, distinguished speakers, and esteemed attendees. It is my privilege to deliver the vote of thanks at the International Symposium, Chemistry for a Sustainable Future, Bridging Science and Society. At the outset, I extend my deepest gratitude to our revered founder chancellor, Bhagwan Sri Sathya Sai Baba, whose vision and grace have been the guiding force behind this symposium's success. His inspiration continues to illuminate our path towards excellence and service. I express my heartfelt thanks to our administrative leadership, Sri Chakravarti Garu, Honorable Chancellor of the University, Professor Raghavendra Prasad, Vice Chancellor, Dr. Shrikant Khanna, the Registrar, and Professor Pallav Kumar Barua, the Dean of Sciences, for their unwavering support and strategic oversight, which were instrumental in ensuring the seamless execution of this event. Our deepest appreciation goes to the distinguished speakers who have shared their expertise and insights on sustainable chemistry. Your thought-provoking contributions have enriched our understanding and inspired meaningful dialogue on the intersection of chemistry and sustainability. To our enthusiastic attendees, scientists, educators, students, and industry professionals, thank you for your active participation and engagement. Your presence and interactions have significantly elevated the intellectual discourse of this symposium. A special acknowledgement is due to Professor C. Sundareshan, whose support and encouragement have been pivotal, as well as Professor Chelli Janardhan and Professor Nageshwar Rao for their invaluable guidance. I extend my sincere thanks to the faculty and the leadership of Department of Chemistry, particularly Professor Rajini Bandari, convener and head of the department, Dr. Ravi Kishore, co-convener, and Dr. Kumar Sai Smaran, organizing secretary. Your meticulous planning, dedication, and attention to detail have been the foundation of this event's success. I would also like to acknowledge the faculty in charges of various committees and dedicated student volunteers whose tireless efforts ensured every aspect of the event was executed with precision. From the vibrant rangoli and the elegant menu decorations to seamless arrangements for accommodation, hospitality, and the delicious food and refreshments provided throughout both days, your commitment behind the scenes has been truly exemplary and instrumental to the success of this symposium. Our gratitude extends to the technical and the audiovisual team led by Sri Raghuram for ensuring a smooth and engaging experience for all participants with your precision and professionalism. As we conclude this symposium, let us carry forward the ideas, knowledge, and collaborations fostered here today. The journey towards sustainability is a shared responsibility, and I'm confident that the inspiration gained here will translate into tangible contributions towards a more sustainable future for chemistry and society at large. With these few words of gratitude, I formally conclude the vote of thanks for this session. Thank you all, and may we continue to work together for a brighter, sustainable tomorrow.
We shall now conclude the session with Mangal Aarti to Bhagwan Sri Sathya Sai Baba. I request the dignitaries to kindly stand for the Aarti. Thank you.
जय जगदीश हरे स्वामी सत्य साई हरे भक्त जना संरक्षक भक्त जना संरक्षक परति महेश्वरा ओम जय जगदीश हरे शशिवदना श्रीकरा सर्वा प्राणपते स्वामी सर्वा प्राणपते आश्रित कल्पलतिका आश्रित कल्पलतिका आपद बांधवा ओम जय जगदीश हरे माता पिता गुरुदेवमो मरियंतयु नीवे स्वामी मरियन्तयु नीवे नाद ब्रह्म जगन्नाथ नाद ब्रह्म जगन्नाथ नागेन्द्राशयना ॐ जय जगदीश हरे ओंकार रूप ओजस्वी ओ साई महादेवा सत्य साई महादेवा मंगलारति अंदुको मंगलारति अंदुको मंदर गिरिधारी ओम जय जगदीश हरे नारायण नारायण ओम सत्यनारायण नारायण नारायण ओम नारायण नारायण ओम सत्यनारायण नारायण ओम सत्यनारायण नारायण ओम जय सद्गुरुदेवा नारायण नारायण ओम सत्यनारायण नारायण नारायण ओम नारायण नारायण ओम सत्यनारायण नारायण ओम सत्यनारायण नारायण ओम जय सद्गुरुदेवा नारायण नारायण ओम सत्यनारायण नारायण नारायण ओम नारायण नारायण ओम सत्यनारायण नारायण ओम सत्यनारायण नारायण ओम जय सद्गुरुदेवा शांति 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 समस्त लोकाः सुखिनो भवन्तु समस्त लोकाः सुखिनो भवन्तु समस्त लोकाः सुखिनो भवन्तु ॐ शान्ति शान्ति शान्तिः जय बोलो भगवान श्री सत्य साई बाबा जी की जय।